I'd like to go over just a couple more topics for chapter 11 that we didn't get a chance to really talk about since we didn't have lecture and they weren't specifically covered on the worksheet. So the first one is shuttle systems that determine the net ATP produced from the oxidation of a molecule of glucose. So if you look at the table in your textbook, it tabulates how much ATP you make, and it depends on the type of cell. So here we have liver and here we have muscle. A lot of cells behave like liver. Muscle is kind of more unique. So this is mostly important in the muscle. This is important in most of the other cells. So what we have here is a tabulation of ATPs made. And over on the left-hand column, we have the reactions. So the first box is all the glycolysis reactions. The middle here is the PDH reaction that takes pyruvate and makes it into acetyl-CoA. And then in the pink box, we have the citrate cycle reactions. And then the very bottom is the conversion of those reduced cofactors to ATP in oxidative phosphorylation. So glycolysis yields us a net of 2 ATPs. Then in the citrate cycle, we make a net of 2 ATPs with the GTPs that are directly made in the succinyl-CoA synthetase reaction. These are energetically equivalent to an ATP, so they count as 2. And then all those reduced cofactors are converted to ATP in oxidative phosphorylation. We get 2 NADHs from glycolysis, we get 2 from the PDH reaction, and then the rest are from the citrate cycle. So if you look at conversion of 8 NADHs in the mitochondria, you get 20 ATPs, because remember, for every NADH, we get 2.5 ATPs, and that will equal 20. 8 times 2.5. For FADH2, it's times 1.5, so you get 3. So these numbers are all the same. It doesn't matter what tissue type. The only difference is here. So in the liver, you get 5 ATPs, and in the muscle cell, you get 3, and that's dependent on what type of shuttle system gets the NADH into the mitochondria. So we'll look at that now. The first one is the one that occurs in the liver, and that's called the malate aspartate shuttle. So there's a lot going on in this picture. All you need to focus on is the sort of the right half of the picture. So here's glycolysis. It generates NADH. This is in the intermembrane space. So the NADH can get into that part of the mitochondrial membrane structure. That NADH is converted to NAD+, so it's an oxidation, and the concomitant reduction is oxaloacetate to malate. Malate can actually get into the matrix because it has a specific protein transporter. So the malate will go into the matrix, it'll get reoxidized to oxaloacetate, and there is a corresponding NAD+ reduction to NADH. So then this NADH goes through electron transport or oxidative phosphorylation and makes your ATP. So it's a 1-for-1. For every one out here that makes a malate, you get an NADH inside and it makes ATP. The NADH here is not the same molecule of NADH here, but the reducing equivalents are the same. Now for the muscle cell, it's a different shuttle. It's called the glycerol 3-phosphate shuttle. And in this shuttle, again, we have glycolysis here. Here's the cytosol. We take the NADH generated in glycolysis, oxidize it to NAD+. But here, instead of making malate, we make dihydroxyacetone phosphate into glycerol 3-phosphate. Then that will go through a transporter in the outer mitochondrial membrane. The glycerol 3-phosphate then will be used by this dehydrogenase, reoxidized to dihydroxyacetone phosphate, and the corresponding reduction is FAD to FADH2. So then this FADH2 can be used by oxidative phosphorylation and make ATP. But remember, if you're starting with FADH2, you get less ATP than you do if you start with NADH. So this one's not a 1-for-1 because we're starting with NADH from glycolysis, and we end up with FADH2 and oxidative phosphorylation. So that's why you get less. So again, what I was trying to explain is if you start with NADH in the liver or kidney cells, it's a 1-for-1 and you get the full complement of ATP. 2 times 2.5 is 5. If you're in the liver cell, instead of the NADH from glycolysis going on, it gets converted to FADH2 instead, and you get less ATP generated. So that's why it's 32 versus 30 ATPs, depending on what tissue type you're starting with.